Example of a controlled experiment. Hypothesis. If people chew gum, it will improve their memory. Independent variable. Chewing gum. Some people will chew gum, some will not. Dependent variable. Memory. All groups should have their memory checked both before and after the experiment to see if it was improved. Control group. Doesn't chew gum. Remember, the control group never receives the new treatment. Experimental group. Independent variable. Group that chews gum. Constants. Should be the same for both groups. People in each group should be of similar health, with similar memory, with similar mixes of sexes, ages, and ethnicities. Each group should also be tested in the same way. Data collected. You should test people's memories both before and after the experiment. 4. Characteristics of a good experiment. A. Can be repeated the same way and get the same results. B. Have large sample size, many test subjects. C. Are performed over longer periods of time. D. Test only one independent variable. All other characteristics of the tested groups should be the same. E. Are peer-reviewed, examined by other scientists to determine its accuracy. F. Must test the hypothesis and show whether it is right or wrong. G. Is objective. The experiment and conclusion are fair and unbiased. Fact and opinion are not mixed. H. The experiment follows established ethical and legal standards. 4 required Regents labs. 1. Diffusion through a membrane. Part A. 1. What you did. Made a model cell using dialysis tubing. Put glucose and starch inside your cell. Put starch indicator, iodine, outside cell. 2. What you saw. Inside of cell turned black because iodine diffused into the cell. Because outside of the cell was not black, you know the starch did not diffuse through the membrane. Used blue glucose indicator, Benedict solution, to see that glucose did diffuse through the membrane. 3. What you learned. Small molecules, glucose, iodine, can diffuse through a membrane on their own. Large molecule, starch, cannot diffuse through a membrane on their own. You can use indicators to identify the presence of specific substances. Part B. 1. What you did. Looked at red onion cells under the microscope. Added salt water to the onion cells. Added distilled, pure water to the onion cells. 2. What you saw. Salt water caused the onion cells to shrivel. Distilled water because the cells caused the cells to swell back to normal. 3. What you learned. Salt water causes water to diffuse out of a cell. In pure water, water will diffuse into a cell. 2. Beaks of finches. A. What you did. Use different tools to represent different finch species competing for food. B. What you learned. Different environmental conditions, food, favored different species of finch, allowing some to survive and reproduce, but not others. 3. Making connections. The Clothespin Lab. Part A. 1. What you did. Measured how exercise, jumping jacks, affected pulse rate. 2. What you learned. Exercise increases pulse rate. Part B. 1. What you did. Squeezed a clothespin for 1 minute, then squeezed it again for another minute. 2. What you learned. If you squeezed less the second round, it may have been because your finger muscles were fatigued because of lactic acid buildup, waste product of fermentation. If you squeezed more the second round, it may have been because your finger muscles were warmed up from increased circulation. Relationships and biodiversity. Botana Curis Lab. What you did. Compared 4 species of plants based on structural, physical, and molecular, chemical and genetic traits. What you learned. 1. Species that are related share similar traits. 2. Different techniques, such as gel electrophoresis and paper chromatography, can be used to determine relationships between organisms. 3. Endangered species should be protected because they may offer benefits to humans. Gel electrophoresis, a technique used to show how species are related to one another. Restriction enzymes cut DNA into fragments, which are placed into a well in a gel plate. An electric current carries the DNA fragments through the gel, from the negative pole next to the labeled well to the positive pole. The DNA fragments separate according to size. Smaller pieces of DNA are carried farther from the well than larger pieces. This creates a pattern of bands which is unique for every organism. Related organisms show similar banding patterns because their DNA has similar length base sequences.